For the model of the DNA structure, we choose the materials of foamy, toothsticks, and clay for kids, obviously. It's quite fragile, but it's simple. We There was cut for 16 pieces of pentagon-shaped red foamy to represent the sugar, uh, 8 yellow hexagons to represent the pyrimidines, and also 8 um, yellow foamy hexagon shaped plus pentagon shaped together to form the purines, that the and little clay balls for the phosphate. And then obviously the toothpicks were the ones that connected everything and represented the hydrogen bonds between the pyrimidines and the purines.
Okay, now, the reason why we selected those materials it would be because they are easy to obtain and manipulate. Not to mention that the price is also really cheap. Not only that, but they help build a simple model with simple materials, which was a very efficient way of making the 3D model.
DNA is vital for all living beings, even plants. It is important for inherence, coding for proteins, and the genetic instruction guide for life and its process. DNA holds the instruction for an organism or its cell's development and reproduction and ultimately death. DNA is linked with the proteins because DNA carries the codes for proteins. However, the actual protein differs a lot from the codes present on the DNA. The basic steps include the transcription, the translation, the modification and folding, the coding for proteins, the DNA replication, and the DNA inheritance.
For the DNA model, we use some clay and moldable foam. For the phosphate backbone, we use some foam, and for the adenine, thymine, cytosine, and guanine, we use some clay. Also, to be able to stick the clay to the foam, we use some wooden sticks so that the model was so much more stable and the clay could stick to the foam and didn't fall out.
I am Miranda Morales, and I'm going to explain how I did the 3D model of the DNA. First, I made the sugar phosphate backbone with modeling foaming. I rolled them to make 2 equal strips. Then, I made the bases with the forms of tubes, each one with the same length, and I chose different colors for each base: cytosine, thymine, adenine, and guanine. Then I joined the bases of clay, made of clay. I made them of clay. I joined them with toothpicks to the foamy backbone. Since this was really fragile, I had to reinforce the structure with cold silicone. When the bases were added to the sugar backbone phosphate of foamy, I glued them to 2 plastic cups with hot silicone so it will dry faster. Then, when they were well dried and didn't move, I trailed them to make the helix shape. Finally, I glued the plastic cups to a wooden surface so that it wouldn't move and stay like that. And I added the marks on the structure of which part was what. The model is, since it's made of modeling foamy, it dries very slowly, so it is very fragile, but it can stand up really well.
Well, we decided to make this 3D model because it seemed really fun and interesting to make. But we had a couple of problems while doing it. For example, the elongated and thin nature of the overall structure make it really weak and fragile. For example, the bases were too weak to be properly maneuvered, so we had to change them completely. And the overall structure needed a lot of time to fully solidify into one piece that could be easily maneuvered.